Good afternoon. Welcome to our Connect Science seminar. My name is Min Tam, or you can call me Tommy. I'm a senior scientist in Dana Farber. On behalf of Shirley Liu, I will be the host today. So first of all, I want to thank Claudian for organizing it. It is my honor and pleasure to introduce Andrew Gentles, our speaker today. Andrew and I both work on a big consortium project called Cancer Immune Monitoring and Analysis Centers and Cancer Immunological Data Commons project as part of the NCI Cancer Moonshot Initiative to maximize the potential of immunotherapy treatment for cancer. Andrew co-lead the Stanford Cancer Immune Monitoring and Analysis Center, and I am leading the bioinformatics effort for the data commons. So it's really nice to see Andrew again here. So Andrew has a very unique background. He got his bachelor degree in physics at the University of Manchester, UK, and a PhD degree in theoretical physics at the University of Southampton. He then pursued 2 postdocs at the University of Nottingham and Stanford in evolutionary genetics and mathematics, respectively. He then was appointed as an assistant professor in the Department of Medicine and the Biomedical Data Sciences at Stanford University. His main interests are in computational systems biology, particularly the integration and analysis of different types of data, such as genomic data and the clinical outcomes. His group has expertise in statistical and bioinformatic methods for analyzing genomic data. So for example, CyberSort is one of the most popular immune deconvolution tools that Andrew helped to develop. So at the end, we will take questions. Please write questions in the Q&A tab, and I will moderate the questions in the end. Today, Andrew is going to talk about computational identification of cell states and ecotypes associated with outcomes in solid tumors. So the stage is yours, Andrew, take it away.
Thanks, can you hear me okay?
Yes.
Okay, great. So thank you again for the invitation. So I'm just gonna switch to my other title slide, which shows where I am at Stanford. I'm part of the Department of Medicine and specifically the Biomedical Informatics Research Division of the Department of Medicine at Stanford. So what I'd like to tell you about today is some work which actually just came out a couple of weeks ago. It was published in Cell recently about identifying cell states and what we call ecotypes associated with outcomes across solid tumors. So this is work which was done by 2 postdocs, Bogdan, who's in my lab, and Chloe, who's in Aaron's lab. And this was a close collaboration with Aaron Newman, who, as Tommy alluded to, I've worked with quite closely over the last 5 or 6 years. And so that collaboration really started when I was developing a resource called Precog, where we were bringing together all the cancer gene expression data sets that were available in the public domain, where we also had clinical outcomes such as survival times, and we were looking at the association of things like gene expression with survival outcomes. And around the same time, Aaron had started developing CyberSort, which is a deconvolution algorithm. And so we started collaborating really to combine the 2 tools. So using CyberSort to deconvolve Precog so that we could understand something about not just which genes influence survival outcomes, but also what cell types, and particularly what immune cell types. As you know, there's a lot of excitement about immunotherapies these days. And so that was about 5 or 6 years ago. And over the last couple of years, we've been working on the new resource called Ecotyper. So if you're familiar with the history of CyberSort, you'll know that originally it was developed as a deconvolution platform, which can basically estimate the proportions of different cell types in a gene expression bulk profile. So if you have a gene expression profile of a tumor, then CyberSort can tell you what the relative proportions or fractions of different cell types are in that particular bulk profile. And over the last couple of years, what's developed is CyberSort X, which is the ability to actually not just infer the fractions of different cell types and mixtures, but if you have enough data, you can actually at least partially reconstruct their gene expression profiles within the mixture. And so what we've been doing in the last couple of years is basically to extend CyberSort X together with integrating it with TCGA and Precog to develop a new tool called Ecotyper, which is what I'm going to tell you about today. So as everybody here knows, the tumors are very complicated, really ecosystems. So this is a slice through a breast tumor where the nuclei of the cells are stained with DAPI in dark blue. And so you can see it looks like a relatively homogeneous sheet of cells in this slice. But if we start to bring in markers for specific cell types, for example, the malignant cells, we can see that there's actually a lot more structure to this tumor. It's really a kind of, it's really a tissue in itself still. So there are malignant cells, there are different types of lymphoid and myeloid cells, and how the tumor behaves both biologically and in terms of its response to treatment and how this patient responds depends on, what cell types are here, what transcriptional state they're in. We're beginning to appreciate that there's also an influence of how they're actually organized spatially with respect to each other. So it's becoming really important to try to understand these things in more detail if we want to be able to predict who's going to respond to immunotherapy and to try to develop new potential targets for therapies. So it's not just a question of whether the tumors, people have started to refer to tumors as T-cell infiltrated as hot tumors and T-cell excluded tumors as cold tumors, but we're beginning to appreciate it's not just T-cells, it's not just the infiltration, it's the state that they're in, and it's also their organization and their relationship to other cell types as well. So what existing technologies do we have to try to dissect the tumor microenvironment? Well, we have some of the classical methods like flow cytometry and IHC, and they're great because they're very they're very cheap. You can do a lot of samples quickly, and so you can look at large cohorts, but you can only look at a relatively small number of markers simultaneously. So maybe a couple for IHC, and if you're a ninja with the flow cytometer, you can maybe get up to 18 colors and look at 18 different cell types. But there's still a limit in terms of the number of things that you can look at simultaneously. And of course, in recent years, we're developing technologies such as single-cell RNA-seq, where in principle, you can get a completely unbiased look at what's in a biopsy of a tumor, for example. But in practice, we know there are still some distortions. So there's still some processing that you need to do in terms of dissociation of things. And so certain cell types get lost during that process. So we know, for example, that plasma cells and granulocytes tend to be preferentially lost during dissociation and flow sorting. And then just the act of actually performing these operations on cells, so sorting cells out, if it's a fibroblast, you may need to do a mechanical and enzymatic digestion. So these cells may be sat around for quite a time, sat in enzymes or having some mechanical process, and that tends to distort their gene expression profiles. And so we know there are certain biases which come with single-cell RNA-seq, even though these are improving rapidly. And of course, it's still relatively expensive, and it's hard to ramp this up to hundreds or thousands of samples in order to make inferences about treatment responses. So we've really taken the tack of trying to exploit some of the bulk data sets that are out there. So over the last 20 years, there have been literally, I think there's over 1 million gene expression experiments now deposited in GEO. So there are these large cohorts of cancer data sets, and if you're interested in other diseases, of course, many other types of cohorts. But the nice thing is they often have very rich clinical outcomes, but of course they're all from bulk biopsies. And so if we want to try to understand something about cell-specific effects from these data sets, then we need methods for actually inferring something about cell-specific transcriptional profiles from the bulk expression data. And so that's where CyberSort and other deconvolution methods come in. So the idea is that if we have a biopsy from something like a diagnostic tissue from a tumor, that's a bulk tissue, which basically gets blended up. We do some genomic profiles, such as gene expression profiling, and then there are various methods such as CyberSort or Excel and Timer, which can estimate different cell fractions that are present in that mixture. That's been the status for a while now. There's various methods that are out there in the literature. What we've done in the last few years is to take that one step further to try to, as I say, reconstruct the gene expression profile of individual cell types within mixtures. So what do I mean by that? Essentially, if we have a cohort such as a set of cancer patients, so you would typically have tens, hundreds, maybe even thousands of samples in the columns here, and you measure the gene expression, for example, in the rows. But we know that each sample that we have, this gene expression profile is a mixture of different cell cell types in that mixture weighted by their relative fractions. So that composite gene expression profile is some fraction of these different cell types multiplied by whatever their gene expression profile is within that mixture. And so what CyberSort-X tries to do is basically to try to take the mixed profiles and to actually invert that and learn something about the cell type specific gene expression profiles. So you'll know if you're at all familiar with linear algebra, that's a very underdetermined problem. So in principle, the gene expression profile of a T cell could be different in everybody's tumor. In practice, we make, of course, the assumption that's not true, because if a cell state is unique to every patient, then we're going to face a really uphill battle in trying to learn anything about the disease. And we know from past experience that there are shared things in cell types across, responders and non-responders, for example. But mathematically, we can express this problem as saying that the mixture profile on the right here, the M, is a linear combination of the cell type specific gene expression profiles weighted by whatever the fraction of the different cell types is in the mixture. And so, as I said, in principle, this H matrix could be different for each patient. But in practice, we make the assumption that there are certain things which are conserved. And so in particular, if we want to estimate the fractions of different cell types, one thing we can do, which is to make a reasonable assumption, is that there are some genes which are essentially sort of marker genes or barcodes for specific cell types. So CD4 T cells, for example, we know at the protein expression level that they express CD4 on their surface, and that's how we use, one of the ways we use to isolate them with flow cytometry or in CyTOF. And so we make the assumption that, a CD4 T cell, regardless of what it's doing and what state or condition or tissue it's in, there are certain features of CD4 T cells which are conserved for all CD4 T cells. So whether it's in the blood or in somewhere in a tumor, we assume that there's some set of genes which are characteristically expressed by CD4 T cells and other types of cell. And so if we actually take that subset, so instead of looking at all the possible genes which are expressed on different cell types, we look at the ones which are characteristic of specific cell types, and we refer to that as a signature matrix. Then we can quite easily, if we have a bunch of expression profiles from mixed samples, and we have a signature matrix which we've defined somehow, then we can easily estimate the fractions of the different cell types that are present in each of the individual samples. And so then actually what we can do is turn around and then flip that equation on its head, because if we assume that there are also other shared characteristics of the different cell types across particular groups of patients. So let's say, for example, that we have some group of patients within this cohort who are responders to a treatment versus not responders, then we can make a reasonable assumption that there may be something conserved about the CD4 T cells in the responders versus the non-responders. We don't necessarily need to know exactly what gene is being expressed in every single patient in the CD4 T cells. And so if we take the fractions which we just estimated for using the signature matrix, then if we have a particular group such as responders or non-responders, we can simply take the responder bulk gene expression profiles, stick in the fractions for the cell types, and we'll actually get an estimate of kind of what the average CD4 or CD8 T cell looks across responders. And we can do that for another group such as non-responders, and then we can make comparisons between them. So that's what we call group mode in CyberSort-X. And then the capability it has on top of that is to actually extend this. I'm not going to go into the details of how it does it, but it's essentially a sliding window approach, is that we can actually infer the gene expression profile of specific cell types at the individual sample level. And it's not perfect, so we don't get the whole transcriptome. The number of genes that we can reconstruct for any particular cell type depends on various things, obviously the strength of their unique signature, how represented they are within the mixed sample. So if you have something which is only there at like 1 in a million cells, then you're not going to be able to reconstruct much about its gene expression profile. But you can imagine that if it's 1, 5, 10%, or even higher of the cells, that we can actually often reconstruct the gene expression profile for up to thousands of genes in any particular cell type. So how do we get this signature matrix? So typically in the past, what we did was to take gene expression profiles of sorted samples. So if you're familiar with the original version of CyberSort, you may have heard of a signature matrix called LM22, which we used in the PRECOG study, and that was based on 22 different immune cell types that were actually isolated from primary blood and then had gene expression profiling. And so we were able to define essentially a signature matrix for those 22 different cell types. But in principle, you can get it from anywhere. So the newer version of CyberSort X has the capability, for example, to take a single cell RNA-seq data set from tumors. You can do your normal clustering, define markers of the different cell types which are present, and then use that as the signature matrix. So there's various ways of getting hold of this signature matrix. And so the idea is the same in each case, is that we have biopsies from example from a tumor or a blood draw. We do bulk gene expression profiling, and then using the signature matrix, CyberSort X can deconvolve that to estimate the fractions of the different cell types in the signature matrix that are present in the mixed sample. And then, as I said, with the group mode, we can take, two groups, for example, responders and non-responders, and we can split the gene expression profile of each of the cell types into something which is representative of what it looks like in responders or non-responders, and then take it even one step further to do this sort of individual sample by sample reconstruction of the gene expression profile of the different cell types. So just to give you an example of how that works, this was applying CyberSort X to the lung cancer data that are part of TCGA. So there's actually two types of lung cancer in TCGA. There's adenocarcinoma and squamous cell carcinoma. So basically what we did was to apply the high resolution purification capability of CyberSort X to the lung cancer samples in TCGA. And so we basically reconstruct the gene expression profile of the epithelial, the immune, and the fibroblast cells. We also did the endothelial, but there wasn't a difference between adenos and squames, which is why I'm not showing it here. But basically, we reconstruct the gene expression profile of the epithelial cells, the immune cells, and the fibroblasts across TCGA. And then when we do some clustering on it, we actually see that we pull out the two different types of epithelial cells in adeno versus squame. So these are genes which are typically expressed in these different types of lung cancer, for example, the basal keratins and TP63 in squamous cell carcinoma. But interestingly, we also found that there were some differences in the immune populations as well. So we saw there were genes which were within the pan-immune CD45 plus compartment were differentially expressed between the adenos and squames, and similarly to the fibroblasts. And this is done without any reference to the actual histology labels in TCGA. So CyberSort X knows absolutely nothing about the existence of adenocarcinoma versus squamous cell carcinoma, but it's able to find these differences between them in these different compartments. And it turned out that around the same time, we'd actually been doing a project where we were actually sorting different cell types from lung tumors, both adenos and squames. And so what we could do is to actually compare this directly to ground truth data. So on the right here is showing the same genes in the rows as on the left, but now I'm showing the gene expression profiles of these different cell types that we had actually sorted from tumors. And so you can see it quite nicely recapitulates the findings from the deconvolution approach. So what we did subsequently is basically to kind of crank this up on a much wider scale, following the sort of study that we'd done previously with PRECOG. And so we've developed this resource and tool that we call Ecotyper. So basically what we've done is to systematically apply deconvolution across most of TCGA. For various reasons, we decided to focus on the solid tumors, the solid carcinomas. So there's 16 of those in TCGA out of the 33 total cancer types. We have a total of about 6,500 samples. So they're mostly bulk primary tumors. There's about, I think, 500 or so adjacent normals. And so what we do is to systematically deconvolve TCGA with CyberSort. And so as an initial starting point, we take a sort of signature matrix which has 12 different cell types in it, which we think are representative of what are likely to be present in the solid tumors. So various types of immune cells, but then also fibroblasts and endothelial cells, and obviously epithelial cells. So basically, CyberSort X estimates the fraction of those cell types in each of the 6,500 samples from TCGA, and then we take it a step further and try to reconstruct the gene expression profile of those cell types within each of those individual samples. I'm sorry about the construction site, which is just outside the window. So, obviously, when you do that, any particular individual patient sample may not just be one unique type or flavor of CD8 T cell or CD4 T cell or monocyte macrophage. There may be a mixture of different profiles within that. And so we actually do a second subsequent level of deconvolution using non-negative matrix factorization, or NMF, to actually try to split the CD8 T cells into, for example, sort of different baseline expression profiles, which are replicated across cancer types. And I'll show you some of the results of that coming up. And as I mentioned, this work just recently came out. Bogdan and Chloe are the joint first authors, and it's out in Cell about a week or two ago. So then what we can do is, once we've got these cell type specific gene expression profiles, we can start to do things like clustering. So we can look to see, are there different transcriptional profiles of fibroblasts or CD4 T cells which recur across different cancer types or within a specific cancer type, for example. So this is showing you how this works. And so this is actually the raw output from CyberSort X from the high-res purification. So we have our 12 different cell types here. And basically what we get out of CyberSort X is a heat map for each of those different cell types. So we've gone from one heat map of the mixed gene expression profile with 6,500 columns and 50,000 rows or whatever it is, depending on how you did the gene expression normalization. And we end up with a decomposition of that into a heat map for each of the 12 different cell types. And so in each of these heat maps, there's 6,500 columns again. As you can see, the number of rows differs. And that, as I mentioned, that's depending on the prevalence of the cell type and how strong their gene expression profile is, dictates how many genes you're actually able to reconstruct for each of them. But given these, what we can then do is to apply this NMF approach to define clusters. And this is showing you exactly what happens when we apply that clustering. So you can see that each of the heat maps actually organizes quite nicely into different transcriptional states, which we think correspond to different phenotypic states of these different cell types across TCGA. So if I show you a specific, I'm going to come to a specific example in a second. But just to give you an idea of how that looks in terms of the number of different cell states that we find for different cell types, these UMAP plots on the right here are showing all of the TCGA samples. So each of the points here in each of these UMAPs is a sample from TCGA. And it's just showing you for each of those samples what the prevalent transcriptional state of the different cell types is within that sample. So obviously, each sample could have a mixture of different types of B cells, but there's going to tend to be a particular type of B cell which is highest in any individual tumor. And so what you can see is, if we look, for example, at CD8 T cells, you can see there's a green, a red, and a blue, corresponding to the fact that we discover 3 different flavors of CD8 T cell, but it can go up to anything like 9 different states for the monocytes and macrophages and dendritic cells, for example. We also see that there's some enrichment for normal adjacent states. So if I toggle backwards and forwards here, you can see that there's this purple state of B cells here, for example, and that tends to be most prevalent within adjacent normal samples, for example, and we see that for other cell types as well. We're not just discovering cancer-specific states. So if I actually color the points by which histology they are, you can see that they're basically scattered. So it's not that the different states of B cells that we find correspond to, B cells in breast cancer versus B cells in ovarian cancer and so on, they actually look like they recur across the different cancer types. And that's partly because of the way that we set up the analysis. We were specifically looking for things which recurred across cancers rather than doing a very cancer-specific analysis, which is something we're doing in parallel. So this is just to summarize where we've got to so far with ecotypes. So the first step is essentially CytoSort X, where we take our bulk gene expression profile, we decompose it into fractions and then to cell type specific gene expression profiles. The next step is to actually take these digitally purified profiles, for example, of CD4 T cells, and to decompose that into kind of what their basic states are that recur across tumors, and then to actually weighted by their representation in any particular sample, and that recapitulates the original gene expression mixture for that particular cell type. So this is giving you an example of what happens with fibroblasts, for example. So I'm just taking this heat map here and kind of expanding here. So for the fibroblasts, we find 8 different transcriptional states. As you can see, there's this particular state here, state 2, which is enriched in the adjacent normals, which are indicated by these blue bars in this kind of salmon-y strip here. And then we can start to do things like put labels on these based on the specific genes that they're expressing. So we've gone through and annotated a lot of these different cell types. So for example, in the fibroblasts, we recapitulate 2 specific types of cancer-associated fibroblasts, which were originally described in head and neck cancer by Purham et al. A few years ago in Cell. With a single cell study, they found that there were these 2 different states of fibroblasts, which they called CAF1 and CAF2. We've subsequently also found evidence for them in other cancer types like lung cancer, but with this ecotyper approach, we actually see that they recur across many different tumor types. And we can go on to try and put labels on some of these other types of fibroblasts as well. So we find a myofibroblast-like state, a couple of migratory types of fibroblast. Interestingly, these sort of reflect different aspects of migration. So this migratory 1 type fibroblast is one which expresses genes which have been shown to be expressed in fibroblasts, which themselves are kind of moving and migrating around, whereas this migratory 2 is actually genes which overlap with some previous data suggesting that there's a particular type of fibroblast which secrete things which then cause epithelial cells to migrate around. So we have these 2 different aspects of migration related to fibroblasts. One's intrinsic and one is a sort of effect telling epithelial cells in essence to move around and to disconnect from the local ECM. Again, for macrophages, we can do the same thing. So for macrophages, we find 9 different transcriptional states. A couple of them are enriched in the adjacent normals. If we start trying to put labels on them, we're able to find, for example, classical monocytes, but also M0, M1, and M2 type macrophages. And this is kind of interesting because there's still some controversy, I think, in the macrophage literature about whether these M0, M1, and 2 actually correspond to physiological states, because typically they're defined by taking undifferentiated macrophages and causing them to polarize under different media conditions into these M1 and M2 states. And so I think, it still wasn't entirely clear with how closely they correspond to actual physiological states in the body, but we're actually able to recapitulate those from deconvolution of primary bulk tumor samples. Interestingly, we also found this particular type of macrophage, which we labeled as foamy macrophages, because transcriptionally they look very similar to macrophages which have been previously described as involved with atherosclerotic plaques. So you get buildup of cholesterol in the arteries, and you tend to get accumulation of this particular type of inflammatory macrophage called foamy macrophages. But to our knowledge, we hadn't seen that to be systematically described as associated with cancer in a significant way, although there were some scattered reports. So I'll come back and show you a little bit more of the data about that shortly. This is just showing how we look at the enrichment to identify which of these previously described macrophage states correspond to our gene expression profile. So we're just basically doing a gene set enrichment analysis to show the enrichment of genes which are specific to these particular macrophages as described previously. So what can we do next? So we've identified cell states, and so then we can do things like look for whether they're associated with things like clinical outcomes and survival, and we can also look and see whether they can be cross-validated in single-cell RNA-seq, and also whether they are associated with specific things like TCR or BCR, diversity, the presence of specific mutations, and activation of particular biological pathways, and so on. So I'm not going to go into all of these details. I'm just going to show you a couple of sort of high-level vignettes of some of the things which we think are interesting from these analyses. And then, of course, we can also we do all the sort of learning part of this on TCGA. We can also cross-validate specifically with precog, which has about 9,000 tumors samples corresponding to the same sample types that we use from TCGA. So we can also do some external validation of these findings. Just to show you very quickly some of the validation in single-cell RNA-seq. So what I'm showing here for each of the cell types, so each of the cell types gets a little heat map, and then just showing you some of the most prominent marker genes for the states that we identify. And so basically what we do is to take these states, we go to the single-cell RNA-seq atlases, and we see if we can assign individual cells to a particular state, and then to see if they actually recapitulate the gene expression profile that we see from the deconvolution. And so you can see with a couple of examples, there's an example here in plasma cells and neutrophils where it's a bit weaker, but generally speaking, we are able to find supporting evidence for these different cell states when we go and look at the single-cell RNA-seq data. I think one of the things that we believe is that our approach is quite complementary to single-cell RNA-seq. So both approaches have pros and cons, and what we found is that we're able to identify some transcriptional states which will validate when we go and look in single-cell RNA-seq, but actually would have been quite difficult to pull out from the single-cell RNA-seq with completely unsupervised clustering if we didn't have some idea of what we're looking for. So as I mentioned, we can look at things like associations with prognostic associations, so specifically overall survival, which is the measure that we typically use in TCGA and also in precog. And so if you're not familiar with it, what we have across here is all the different cell states that we identify. And so if you see a large red bar going upwards, it means that that particular state of that cell type is associated with unfavorable outcome, so shorter survival. And if you see something blue going downwards, it means that that particular cell state is associated with favorable or longer survival. And for the purposes of this result, we've actually summarized across cancer types. So this is basically sort of averaging across the different 16 different epithelial cancer types. Of course, we have the equivalent where we've done it separately in the different cancer types, but then it comes a bit overwhelming to kind of present the association with the outcomes of these 69 cell states across 16 different cancer types. And so it's nice just to have this kind of high-level summary. So we see various things that would be expected or that we've seen previously. So with the CD8 T cells, for example, we actually see a favorable association of this particular state here, which turns out to be, we think, associated with sort of classic cytotoxic T cells, which are actively engaging in killing malignant cells. We've previously seen in lung and breast cancer a very favorable association of plasma cells and mast cells with outcomes. And here we're kind of breaking it down and seeing that it actually looks like it's a specific transcriptional state of these different cell types. Over in the epithelial cells, we see that there are particular states which are associated with shorter survival. And so there's some enrichment for squamous cell tumors there, but also for things like bud tip cells, which are the kind of stem cell like epithelial cells, which may have some association with the differentiation state of tumors. And then some other things which we wouldn't have necessarily expected at all. So if you look for the single most prognostic cell type, there's a particular state of dendritic cells, which is protoplanin positive. And if we look across the whole of TCGA, it's the cell type which we find is most robustly associated with poor outcomes. And we know nothing about this particular dendritic cell state. It's just an observation which we would obviously have to go and follow up on. So actually, but within the monocytes and macrophages, there's this particular state which you can see is associated with unfavorable outcome. It's got this AEBP1 gene expression, and this is the one which I mentioned is, we think, related to the foamy macrophages. And so we wanted to have a little a look in a bit more detail about whether we could validate some of the findings with that. And this is just showing you the consistency with TCGA and precog. So TCGA, we learn the cell states and look at their survival associations on the left, and then we look at the same tumor types on the right in precog and the same cell states. We can apply the NMF framework to estimate their fractions in precog and then to compute the association with survival. And so you can see it's quite consistent between TCGA and precog. So not just spurious results that we're picking up from the TCGA analysis. So as I mentioned, the foamy cell macrophages we found were adversely associated with outcomes, specifically in three cancer types which are driving that pan-cancer association. So colon, ovarian, and stomach. And so this is in the top row showing you the association with outcome in TCGA, which is the training data. And so you can see that the higher the level of foamy macrophages, the shorter the survival is for the patients. And we recapitulate that quite nicely when we look at the same cell types in precog. So you can see again that the foamy cell macrophages are associated with shorter outcomes and worse survival. So we actually wanted to look and see, could we actually, instead of doing this computational approach, which is purely based on gene expression, can we actually then go and look at some samples and say, can we actually see these foamy macrophages sort of in person? And so Matt van der Rijn's lab in pathology here at Stanford had a bulk gene expression profile on some colorectal cancer samples. And so what we did was to apply the ecotype framework to those samples so that we could predict which ones we thought either did or didn't have these foamy macrophages present. And so then Matt's lab, Magda, and in Matt's lab specifically, went back and stained some of the slices that we had made the predictions on. And so this is one example on the left where we had predicted there were no foamy macrophages present, and sure enough, it wasn't possible to find any. But then on the right here was an example of a colorectal cancer sample where we predicted from the gene expression profile that it should have a large number of foamy macrophages. And so these arrows are actually marking some of them. You can see there's these cells here which have a well-defined small nucleus, but then they have a kind of big blobby or foamy cytoplasm around them, and that's the foamy macrophages that we were looking for. So it's kind of a nice validation that we can actually see these cell types. It's not just some computational artifact. So in the last few minutes, I'll talk about how we take this one step further. So instead of looking just at individual cell states across tumors, we can start to ask questions like whether these cell states tend to co-occur with each other. So if a patient has particularly high representation of state 1 of neutrophils, is there some particular state of fibroblasts and CD4 T cells which tends to go with it, for example? And so that's when we started really talking about what we call these ecotypes or tumor ecosystems. So what we're doing is to look for co-association of cell states recurring together across different tumors. So people define ecosystems and ecotypes differently depending on the context. And in particular, I think if you can bring in a spatial aspect as well. And so to talk about whether particular cell types are in proximity and interacting with each other, that's another way of defining an ecosystem or ecotype. Just for the purposes of this, we're looking just at co-association of the presence of specific cell states within particular tumors. So that's kind of like the last step of ecotype here is we we do the deconvolution, we discover the different cell states, which is the second line here, and then we try to find if we can actually assign those cell states or co-assign them to what we call cellular ecosystems or ecotypes. So this is just showing you how this works. So again, this heat map is for all of the solid tumors in TCGA. So each of the columns is one of our 6,500 samples. But now the rows are the different cell states. So there's the 69 cell states which occur across our 12 different cell types. And then the color in the heat map is just the representation of this cell state within each of the samples across TCGA. And so then we can cluster this. And sure enough, we can see that these things tend to occur in blocks. So this top left here, you can see there's I think 1, 2, 3, 4, 5, 6, 7, 8 different cell states, and they tend to co-occur with each other in the same tumors, or to have a high representation in the same tumors. So we basically define 10 of these different ecotypes based on this clustering of the cell state prevalence. As before, we find that we're discovering things which are pan-cancer, which again is partly because of the way that we set the analysis up. But you can see, for example, that breast cancer is the dark blue in this tumor type color bar. And so you can see that it's spread throughout the ecotypes, or conversely, the ecotypes tend to occur across cancer types. So ecotype 1 is not just breast cancer, it occurs in pretty much every cancer type in TCGA, and similarly for the other tumor ecotypes that we discover computationally. We can just represent these slightly differently by these little network diagrams. So this is just showing you that different ecotypes have different numbers of cell types associated with them. So we actually impose a minimum where we're only interested in co-occurrence of at least 3 different cell types. So CE4 with the 3 different cell states going together is the smallest. So there's a couple of orphan cell states which we don't find co-occur with each other in the different ecotypes, but the majority of them occur together with other cell states. And so you can see that they vary in the number of cell types which are present. So we can also look and see whether the ecotypes themselves are correlated with prognostic outcomes, which, as you would expect, so if you would expect that if there's a bunch of cell states in any of these particular ecotypes which are individually associated with outcome, that the E is likely to be associated with outcome. Let me skip across this, and that's true. So you can see there's these 3 ecotypes on the left which are associated with poor outcomes and shorter survival, and these 3 on the right which are associated with longer outcomes, with longer survival and better outcomes. So that's what you'd expect, and it turns out that the cell types or cell states that are present within these ecotypes are the ones which are themselves tend to be individually associated with outcomes. But we see over on the left here where there's this particular group of ecotypes associated with poor outcomes, but then there's a couple over on the right here which are really quite strong predictors of favorable outcomes. And we wanted to understand a little bit more in detail what the composition was of these ecotypes and how they might be influencing the outcomes. So in particular, these 2 ecotypes on the right here, if I go back to the little network diagrams, they're down on the right here. So if you just kind of look at the different cell states that are in here, you can see CD4 and CD8 T cells, macrophages, and so on in both of these cases. So they really look like some sort of inflammatory immune response, which is potentially the immune system engaging with tumors. But even at this level of prognostic association, we can see there's some difference in terms of their association with overall survival. But obviously, based on the fact that these are immune type ecosystems, we were curious to know whether they were actually associated with specifically immunotherapy response. So TCGA, there's all sorts of different types of treatment modalities that patients get, but they're typically sort of traditional types of treatment like surgery and radiation and maybe adjuvant chemotherapy. There isn't much in there in terms of immunotherapy, for example. And there's still relatively few data sets which are available where we have a significant number of samples with genomic data, where we also have things like immunotherapy response. And obviously, this is something we're really interested in as part of the CMAC network. But there are a few data sets out there. So when we did this analysis, these were the data that we had available. And so basically what we did was to take 3 different data sets. So there was 1 in bladder cancer, 2 in melanoma, 4 in melanoma, and using several different checkpoint blockade therapies. And so what we did was to take the gene expression data from each of these studies. We actually used the NMF structure from ecotyper to project our ecotypes onto these new samples. So given a new bulk expression profile data set, you can actually run it through ecotyper and it will spit out what it thinks the representation of both the cell states and the ecotypes is. And I'll put up the website at the end so that people can see if they want to go and run their own data through that. But what was surprising to us is when we looked at the association of basically different signatures, including the ecotypes with immunotherapy response, what we saw was that right at the top of the list was ecotype 9 as being very good at predicting favorable outcomes, both in terms of survival on the left. So this is the survival, so overall survival or progression-free survival, as well as response on the right. So whether somebody achieved a partial or complete response compared to a stable disease or no response. And so, there's absolutely no information about immunotherapy response goes into the whole ecotyper framework with TCGA, but surprisingly, it comes up with this particular ecotype or co-occurrence of different cell states, which is very good at predicting response to immune checkpoint blockade. Actually, even better than the signatures which were actually developed in the papers which published the data themselves. So that was really quite a surprise to find that worked so well. So another thing I wanted to mention is how these ecotypes actually change and evolve with tumor development. So there are various data sets which have a mixture of normal samples and then adjacent normal samples and primary biopsies, for example. And so we can actually look and see how the prevalence of these different ecotypes changes across the evolution of a tumor. And so you can see that on the left here, we really see only evidence mostly for this one particular ecotype, which we had previously referred to as what we call the normal ecotype, because it tended to be most strongly associated with adjacent normal. But you can see that what happens is that's the prevalent ecotype in completely normal tissue from somewhere like GTEx. As you go to things like adjacent normal samples, which come from in the region of a tumor, you can see that there's starting to be somewhat of a shrinkage of this normal ecotype, and we begin to see the emergence of some cancer or tumor-specific ecotypes, as well as some immune response. And then by the time we get to full-blown cancer, you can see that the normal ecotype is largely gone now, and it's been replaced by what we call these carcinoma ecotypes, which are composed at the top, are largely driven by the epithelial cells and fibroblasts, and then also the presence of specific immune responses from these 2 inflammatory type of immune ecotypes. We can see it in another way. There's this nice data set which was produced by Teixeira et al. A couple of years ago, where they had gene expression profiles on pre-malignant lesions from lung, and then they followed these patients over time and to see who progressed to lung cancer versus who had spontaneous regression. And what we found when we projected the ecotypes onto those was that it was the patients who had a skew towards ecotype 9 versus ecotype 10 who progressed to lung cancer versus regressed spontaneously. So for some reason, it looks like you have these 2 inflammatory immune ecotypes, but the presence of ecotype 10 tends to push somebody towards having spontaneous regression of these pre-malignant lesions, whereas a shift towards ecotype 9 is associated with progression to lung cancer. So we wanted to try and understand a little bit more detail about the specifics of these ecotypes, and particularly what cell types and cell states are present in them. So we have all that output. Essentially, we can actually look specifically at the different cell types within these ecotypes and look at what's unique about the gene expression profile of those cell types within them. And so, for example, in the CD8 T cells up in the top left here, orange is ecotype 9 and blue is ecotype 10. And if you're familiar with T cell literature, particularly in the context of immunotherapy, you'll probably immediately recognize that a bunch of these genes, which are highly expressed on these CD8 T cells in ecotype 9, are what we would often refer to as exhaustion markers. Conversely, the genes which are highly expressed on these other CD8 T cells in ecotype 10 are ones which are more commonly you would refer to as being an actively cytotoxic T cell, which is potentially in there engaging tumors or infection and actually having a killing function. And we can also, we can see differences in the other cell types as well, which I don't have time to go into, but there's lots of potential interesting associations here that we can follow up on. So we actually went to, in addition to the fact that there's these two different ecotypes, there's the whole question of, what's different about them apart from the gene expression? Is there something different about the location of the ecotypes, for example? So we actually went back and looked at where some of the specific cell types from these two ecotypes actually occurred. And so I've forgotten the label here, but I think that this top one here is the markers for ecotype 9. So particularly for the T cells in ecotype 9 are granzyme B positive, whereas the T cells in ecotype 10 are granzyme K positive. And then we also looked at the two different flavors of monocytes and macrophages. So in ecotype 9, they were ApoE positive, ecotype 10, they were CCR2 positive. And so we can see that these different cell types tend to co-occur with each other. And we also found interesting that there was a difference in terms of their spatial localization. So what we found was that these ecotype 9 cell states tended to be in the center of the tumor, but not necessarily the edge of the tumor. Conversely, for the other ecotype 9, we actually found that they tended not to be in the center of the tumor, but these cells tended to be around the periphery of the tumor, on the edges. So, this is just a small piece of immunofluorescence data, but we can also look at things like spatial transcriptomics data in a bit more detail. And so we actually took some of the public spatial transcriptomics data that's out there, and we're generating some of our own internally as well. And so if you're not familiar with spatial transcriptomics data, basically this is a slice through, the top is a piece of breast cancer, the bottom is a melanoma. And so this is basically a slice through that. And then what this platform does, which is the 10x Visium platform, is to basically do gene expression profiling on small regions of each of those slides. So it's not quite at single cell resolution yet. In this particular data, each of these dots could have anything from 1 to about maybe 10 cells present. But what we can do is to infer what the most likely ecotype the gene expression in that region corresponds to. And so what we found, which supports the immunofluorescence data, was that you can see that ecotype 9, which is the exhausted one, tends to be infiltrated throughout the tumor. But presumably, it's not, because they're exhausted, they're not actually engaging the tumor. Whereas on the right, we have these actively cytotoxic T cells, but they tend to be excluded around the periphery of the tumor. So it's almost like we have, there's sort of two different conditions here. There's on the right, there's the T cells, which you would like to be in there killing the malignant cells, but for some reason they're being excluded from the tumor. Whereas on the left, you have these T cells, which are infiltrated throughout the tumor, but they're not actively engaging it. And so what we think, we're guessing happens is that when you give the patients with a tumor that looks like something on the left, an immune checkpoint blockade therapy, they suddenly take the brakes off of these T cells, which are exhausted, but they're spread throughout the tumor, and they're able to actively engage and to kill the malignant cells. So I'm going to wrap up there. I hope I've convinced you that we can use deconvolution approaches with large amounts of public data to uncover something about the tumor microenvironment, specifically the different cell states and ecotypes that they form and how those recur across tumors. We think this is really complementary to single-cell RNA-seq and to other methods, and there's a lot of feedback which goes between these different approaches, which I think is a useful two-way thing. And obviously the hope is that this will expand potential targets for therapeutic response and further highlight the importance of studying the spatial architecture. So as I mentioned, this work appeared in the last couple of weeks. So if you want all the gory details, you can find it on that reference. We also have a website called ecotyper.stanford.edu. There's various things that you can do there. If you have a bulk gene expression data set that you want to project our ecotypes on, you can upload it to the website and it will estimate that and give you back the results and some pretty visualizations. If you actually want to run the whole framework, there's a link to the GitHub site there with some very detailed tutorials about exactly step-by-step how you would actually reproduce this analysis or to apply it to a new context with different data, not necessarily even in cancer. We're applying it to other contexts as well, and we hope that it will be generally useful. So I'll end that with acknowledgements. As I said, Bogdan and Chloe really drove this project, and Bogdan in particular came up with the whole NMF-based approach for doing the sort of second level of the deconvolution. Chloe spearheaded a parallel project in lymphoma, which also came out in Cancer Cell a couple of weeks ago. And it's been a very close collaboration with Erin, as usual, and a number of other people across Stanford. And of course, I'd like to thank our funding resources and to thank you for the invitation to talk today.
Thank you, Andrew, for your fantastic talk. Congratulations again for the publication. I'm sure this is going to be a really useful resource for the community. So now we'll open the floor to the audience to ask questions. So we have 2 questions in the Q&A. So first question from Jenny.
Yes, so we're actually in the process of doing that. We've actually built a signature matrix for mouse. We just haven't extensively cross-validated it yet. So we've done some internal computational validation, but what we would like to do is to actually generate some primary data to actually test how well it actually works. So, we've got some single cell RNA-seq from mouse. And so we're going to generate some bulk RNA-seq where we also have some paired sort of ground truth data like codex or something like that to be able to compare them.
Okay, great. Just for the audience who may watch the record, so the question was, have you applied your cell state categorization to mouse tumor models in order to enable study of perturbations on cell state distribution? Okay. Second question is from Raymond. So have you explored ecotyping of cancers with variably associated microbial invaders? This appears to drive epithelial immune inflammatory characters and IO response of a growing number of epithelial cancers.
Yeah, so we haven't looked at that systematically. The one example where we have looked is in head and neck cancer. And so specifically head and neck cancer, there's a particular subtype which is caused by HPV infection, which is a very, and it's associated with particular ecotype and inflammatory immune response. And it's been previously known that the HPV subtype of head and neck is associated with a more favorable outcome. But that's the only case where we've looked at it in detail. That would be a really interesting thing to look at more systematically.
Great. Okay, we have no more questions. I do have several questions. There will be one technical question for the deconvolution of the TCGA RNA expression data. So what kind of normalization do you use? Just because those are different cancer types, how did you do the normalization first?
Oh, before how to bring them together?
Yeah.
So we actually did. So we actually didn't include all of the TCGA data. So we actually did a fairly extensive selection where we did things like clustering everything based on housekeeping genes. And so we found, for example, that there were a few data sets which were just complete outliers when you try to cluster them with the rest of the TCGA just based on housekeeping genes. And then for actually bringing the data together, we actually just use within each histology, we mean, center it, and scale the standard deviation to be
so it's like a Z-score.
Yeah, it's basically a Z-score normalization. Then we bring everything together after we've done some other tests to throw out things which we think are outliers.
Okay, thanks. So the other question is, like, you've discovered several different CAF populations. So I think 2 years ago, there's a paper in Cancer Discovery. They found this called antigen presenting, like cancer-associated fibroblasts. Did you find that as well?
Yeah, so I think you're talking about the paper by David Tuber's lab in pancreatic cancer.
Yes, yeah.
Yeah, so we actually have a project with OHSU, which is specifically doing this deconvolution in pancreatic cancer. And we haven't specifically found that particular type of CAF in the pancreatic data, but we're still in the process of optimizing the analysis specifically for pancreatic. So we're not sure exactly. We find they have the myofibroblast-like state as well, which we actually do find, but we don't. Yeah, we've had some issues reproducing the clustering from the data set.
They also do have this ICAF called inflammatory CAF.
Yeah.
So.
Yeah.
Okay. And you also showed, maybe one last question from me. So you also showed maybe the different subtypes of immune cells, they can locate spatially different in the tumors. So did you like observe that, for example, for M1, M2 subtypes, like whether they are close to the tumor or like they are far away from the tumor?
Yeah, so actually the data I showed with the IF, which I don't think shows up very well on the screen, unfortunately, when it's on Zoom, we were specifically looking at where the macrophages were from those different ecotypes. And it's like one of them was in the center of the tumor, and one of them tended to be around the periphery. So I think what we don't really know much about now is what the nature of these interactions is. It's like, are those macrophages attracting some sort of T cell, or is it the T cells attracting the macrophages? Is there some sort of feedback loop? You know, why they're actually sat on the periphery of the tumor versus inside the tumor, we still don't know. So
is the tumor macrophage, they're more close to the tumor, or they are more far away from the tumor?
Oh, sorry, can you say that again?
The pro-tumor macrophage, are they located close in the center of the tumor, or like outside of the tumor?
So I'm not sure offhand. So the foamy macrophages tend to be in the tumor. I'm not sure exactly with the other types. We haven't systematically done that.
Okay. Great.
There's actually a question from Ankita about whether the NMF code is also available on GitHub. Yes, it is. So basically everything is there. So there's a step-by-step guide to how you would exactly reproduce the analysis that we've done in the paper. So all the R code is there. It has scripts for running it on a cluster. So if you want to scale it up to a big project, that should be relatively possible as well. So everything should be there. And if there's any questions, feel free to get in touch with us, and we'll help out as best we can.
Okay. Any more questions? Okay. So if there's no more questions, we will wrap it up. And thank you again, Andrew, for your presentation. I look forward to seeing more exciting work from your group. And thank you, everybody, for joining us. Okay,
Thanks.
Bye.